Bismillahirrahmanirrahim, 5090, November 24, and we're going to discuss the ATP paper, which was for 40 marks and is for 1 hour. Starting with question number 1, yeast breaks down sugar glucose to provide energy for growth and reproduction by respiring anaerobically. So the glucose is broken down with no oxygen. Anaerobic means no oxygen into carbon dioxide and alcohol. Sugar is used in human food to make it sweet. Too much sugar in the diet can cause diseases. Sugar substitutes are available which taste as sweet as sugar. Like you have stevia and canderel. A student decided to investigate whether yeast can use a sugar substitute for respiration. So the investigation is that yeast can use glucose, but can it use the sugar substitute as well or not? So we don't know. So we're going to do an experiment to find that out. 3 test tubes were set up as shown in table 1.1. There was one with distilled water and yeast and test substance 1 gram sugar. And the other one 15 cm cube of distilled water and 1 gram of yeast and 1 gram of the sugar substitute. And then there was 15 cm cube of the distilled water and 1 gram of the yeast, and nothing was added to it. So one had sugar, one had the sugar substitute, which is stevia, and or we could have used anything else. Whatever was given in the instruction, we would have given that to the students if we were doing a paper 3. And nothing was added. They were just distilled water and yeast. The 3 test tubes were labeled so they could be identified. State the labels. Well, I would just say A, B, C. So I would say just A, B, C test tube containing sugar. This one would have been A, and this one would have been B. And you could have said anything as long as you said something. You could have said sugar S and then SS and then Na. One had sugar substitute, and one had no addition. So you could have come up with anything, but as long as you wrote something, you got this 1 mark. Then it says a stirring rod was used to mix the contents of each test tube. And explain why it's important to clean the stirring rod. Well, of course, because it prevents the transfer of sugar. Of course, if you used it in test tube A, which had sugar, and then you use the same rod to stir test tube B, then what were you doing? You were taking the sugar here and you were adding it to test tube B, because some of it would stick to the stirring rod. So this is something which is very common sense, but of course common sense is the rarest sense. I always say that. So prevents the transfer of sugar to other test tubes. So be careful when writing this. You could have also said prevents transfer of the sugar substitute to the other test tube, which of course had nothing in it. So if you use the same rod in A, which had sugar, and then the same rod in B that had the sugar substitute, and then the same rod in C, well, then you are transferring the sugar and the sugar substitute to the C, which had no addition. Now let's read the rest of the question. The level of the mixtures in each test tube was marked with a water resistance. So this is the starting level. We marked it with a water resistant marker so it wouldn't rub off. And this was the starting level. When the yeast respires, it produces bubbles of gas that are trapped in the mixture, making the level of the mixture rise up the test tube. So you see this froth, this foam sort of a thing being formed at the top of it. And the student used a large beaker as a water bath to control the temperature of the test tube. So these were placed in sort of large test tubes which were used as a water bath. Temperature of the test tubes in this investigation. The temperature of the water at the beginning was 35 degrees Celsius. The aim was to maintain this temperature throughout the investigation by adding hot or cold water. So you had this large beaker and we gave you water at 35 degrees Celsius. Now, after some time, maybe the water was becoming cold. So then you would add a little bit of the hot water. Say it went down to 33. So you added a little hot water to make it back to 35. And if it was too hot, because maybe they didn't give it to you at 35, so then you added a bit of cold water to make it to 35. Then it says the 3 test tubes were placed in the water bath and a timer was started. At 5 minutes, 10 minutes, and 15 minutes. The distance moved by the mixtures from the starting levels were measured and recorded. The temperature of the water bath was also recorded. Figure 1.1 shows the lower parts of the test tubes at 5 minutes and the water bath thermometer reading at 5 minutes. So this is for 5 minutes, please remember that. So starting level, and then you got the temperature, and then you've got the temperature here. You can read off the temperature here. And then it says figure 1.2 show the results of 10 and 15 minutes recorded in the student's notebook. 10 minutes, 31 degrees Celsius, sugar was 38 millimeter, no change in other two test tubes. And in 15 minutes, the temperature was 39 degrees Celsius, sugar was 50 millimeter, and no change in the other two test tubes. So this information is given to you, and now we have to plot this in a table. Part B of the question says complete table 1.2. So we have to complete this table 1.2, complete the headings first. Then using figure 1.1, which was at the back that we had the figure 1.1, this is the figure 1.1. So using figure 1.1, measure the distance moved by the yeast mixtures from the starting level in the three test tubes and record them. Read the temperature shown in the water bath in figure 1.1 and record it. Use the information in figure 1.2 to complete 1.2. So this would be the table. Now the 33 degrees Celsius was this one here. This one here was 33 degrees Celsius. And then of course we had 31 degrees Celsius here. So we were going to give this table here. We were going to just write what was ever here. And then we were also going to give this 38 and 50 here. And the rest were all zero. So you've got to see all these are 0, 0, 0 here, 0, 0, 0 here. And this is the 21, 38, and 50. The 21 millimeter, you measured this. So 21 millimeter was this, and you measured this, and that, of course, would have been, that is why you've gotten this. So this was 5 marks. So headings, time in minutes, and distance for the yeast mixture above the starting level, or you could have said distance in millimeter. And the temperature, please do not write the units. Do not write millimeter here. If you wrote that, that's a -1. Or if you wrote here like degrees Celsius, well, that's again a -1. So we don't want any units. The units are only and only supposed to go in the heading. You can see it's all in the heading. We wrote this in the heading, the units here. We wrote the units here in the headings. And that's the O level rule and that's the A level rule. So it's for everybody, for O levels and A levels. It's the same rule. Units always in the heading. Then it says part 2, explain how the test tube with no addition acted as a control in this investigation, so that a comparison can be made. Control is for a comparison that, okay, there was no sugar, so there's no respiration and no foam or no nothing collects on top of it. And if you added glucose, only then the yeast would respire anaerobically and produce carbon dioxide, and that would result in that foam being formed at the top of it. Then it comes on to part 3. It says state what conclusion you can make from the results of this investigation. It's only 1 mark. Now the conclusion you can make is yeast can only respire using sugar. Or if you said yeast cannot use the sugar substitute uh, to respire. So yeast can only respire using sugar. Or you could have said yeast cannot use the sugar substitute to respire. Then they've asked you part 4, use the information in figure 1.1 and 1.2 to evaluate the student's control of the water bath. Now you can see the water bath is not very helpful because sometimes the temperature was 33, then 31, then 39. So it was not well controlled and the temperature fluctuations were there. So not well controlled and temperature was not constant. Then it says suggest why the temperature of the water bath at 15 minutes was 39. Maybe too much of hot water was added. So too much hot water added could. So the next part, C bar, the yeast activity could also be measured by recording any increase in the volume of the mixture in the test tubes. Figure 1.3 shows the test tube with sugar, yeast, and distilled water as seen from the above. So we've got this test tube and we've got this and we're looking at it from the above. The distance above the marking starting level in the test tube after 10 minutes was 3.8 centimeter. So they've given you this, the length of it. And the line A and B indicates the diameter of the mixture. This line AB is the diameter of the mixture. within the test tube. Measure and record the length of the line between A and B. It was 2.2 centimeter. 2 centimeters given, so I mean, you don't have to write that. 2.2. And now it says calculate the increase in volume of the mixture in this test tube after 10 minutes. Use 3.14 as the value of pi. Give your answer to one decimal place. Show your working. So what have you to do? Pi is, they've given you 3.14. So if the radius, if this is the diameter, then 1.1 is the radius. So into pi r square h. So 1.1 whole square into, and they told you the length of it. The length of it was 3.8 centimeter. So you have to multiply that by 3.8. So the volume increase, you had to calculate that. So the working was like this, and then you answer is 14.43772, but then it said only one decimal place. So one decimal place, you only have to do it up to here. Decimal place, please remember, one decimal place is different than significant figures. Please know that, and you must know the difference between that. So it was 15.4 because the next number is 3 is less than 5. Then it says the apparatus used in this investigation is shown in figure 1.1. Suggest a piece of apparatus that can be used in the investigation to directly measure the volume of the mixture. Describe how you determine the increase in volume at each time interval using this apparatus. You use a measuring cylinder and you subtract the starting volume from the volume at each time interval. So you could have used a measuring cylinder and you subtract the starting volume from the volume at each interval. So if the starting volume was, say, in a measuring cylinder, of course it would be something different. And say this was about 30, and then the final was 45. So 45 minus 30 will be 15. This is what a measuring cylinder looks like, and you of course have them in different diameters. So there are some which are narrower and some which are broader, and of course you have these graduated so you can measure the volume very easily. Now coming to question number 2, when organisms respire aerobically, they use oxygen and produce carbon dioxide. Some students investigated the rate at which germinating seeds respired using the apparatus in figure 2.1. Now you can see there's soda lime to absorb the CO2. They're germinating seeds. That means these are the living cells which are respiring and taking in oxygen and giving out carbon dioxide. Carbon dioxide is being absorbed by the soda lime. Oxygen is being used by the oxygen which is present in the air inside this test tube is being used by the germinating seeds, gauze to support the seeds. We don't want the seeds to fall into the chemical. Then there's a drop of colored liquid, there's a capillary tube, there's a millimeter scale, and this drop is going to move this ways. Why? Because the oxygen is going to be used up and pressure difference is going to be created. And so more pressure will push this drop of colored liquid towards this side, towards this side. Then there's a syringe and there's a three-way tap, because you see you're going to use the syringe to push this colored liquid. When you've done one reading, now you need to reset the apparatus and push it back here. So in order to do that, you use the syringe, and we say reset the apparatus. Please remember to write this, and they usually ask you this question again and again. To reset the apparatus, or you can say move the colored liquid back to the far left, or you say reset the apparatus. Then as we read on, carbon dioxide produced by the germinating seed was absorbed by the soda lime. As oxygen was used, the volume of gas in the apparatus reduced and the drop of colored liquid moved along the capillary tube towards the seeds. The student moved the drop of colored liquid in the capillary tube to the beginning of the scale, 0 millimeter, by opening the three-way tap and using the syringe to carefully push air into the apparatus. They then closed the tap. This was the start position for the drop of colored liquid. Its position on the scale was recorded over the next 4 minutes. The movement of the drop of colored liquid indicates the rate of respiration of the seeds. The students' results are shown in table 1.1. At 0 minutes, the drop was at 0. At 1 minute, it was at 18 millimeter, and then 36, and then 54, and then 72. So this is the time in minutes. You see the heading is always the heading which has, and then the position of the, so it had moved from 0, it had moved to 18, then it had moved to 36, and then it had moved to 54, and then to 72. Then in part A1 it says construct a line graph of the data in table 2.1 on the grid. The values for the endpoints of the axis are shown on the grid. Draw a straight line of best fit to connect the points. The first thing you do is you label the axis. The rule is that the first column, this is the first column, this is the heading. You write this on the x-axis always, that is the rule. First column is always the independent variable and it is always plotted on the x-axis. So you will write here time and minutes, and you will not do any abbreviations, please. Then you will write the next thing. This is position of drop of colored liquid in millimeter will go on the y-axis. This is always the rule. And then you label the axis, position of drop of colored liquid in millimeter, and every 2 centimeter, this is every 2 centimeter, you have to write this 1, 2, 3, 4, 5, and here you have to write 20, 40, 60 because it had said 100 here and it had said 5 here. So you have to spread out the scale, and you should see that you use the whole of the grid as much as possible, the whole of the graph paper. Then you plot the points. I mean, I've written all these points here, and then you plot the points, and either you can use a dot and circle it, or you use a cross. It's up to you. These are the two choices which are available to you. The rule is because how are you going to get 4 marks for this graph? It says both axes fully labeled, linear scale and value at origin of both. So you have to write the 0 here and the 0 here. And so 5 points correctly plotted and points marked with crosses or encircled dots is either this or this. Crosses or encircled dots. If you just made a dot and you didn't circle it, that's a -1. It might be all correct. And then a single straight rule line through all the points. And then you just join them. I always say join it, even if it says line of best fit at times. Here, I can't think we can get a line of best fit. But if you just join it point to point with a ruler, that is the safest bet. So I haven't joined it with a ruler because I can't do it on this thing. So please remember it that you would have to join it from 0 because they was started from 0. But if it doesn't start from 0, if it was only these figures, then please do not connect it, do not extrapolate, do not extend it. So if the 0 was not given, then you should have only started off from the one on which it was say 1 and 8. Then it says use your graph to predict the position of the drop of colored liquid at 5 minutes. Show you're working on the graph, so you have to extend the graph and then use the results at 4 minutes in table to calculate the rate of movement of the drop of colored liquid caused by the respiration of the seeds. Space for working, and that's another 2 marks. So you extended the line to 5 minutes and then you read that off, and that's about 90. So 90 millimeter. So you wrote that here, and because you would show this, of course, how would you get the 2 marks? The 2 marks was the line extrapolated on the graph. So this thing that you did here, this orange line which I've drawn, this would have got you 1 mark, and 1 mark was for the 90, which was reading it off correctly from the graph. Then it says use result at 4 minutes. So at 4 minutes, what was the reading? It was 72. So if you go back to here, 72 divided by 4, because rate means rate of the movement, means per unit time. So it would be 18, but then only there were 2 marks for it. You won't get 2 marks for this. What were you supposed to write to me? You're going to also write a millimeter per minute. So whenever it's 2 marks, remember one would be for this and one would be for the units, because the units were not given here. Of course, here the units was given. But here no units were given. So I'm sure 50% of the students did not write the units, because we now got Gen Z who are extremely careless in their whole the way they do their papers. It's a very careless way that you all do your papers. Some of course you of course are very careful, and I cannot see any careless mistakes in your papers. Then it says part B, plan an investigation to determine the effect of temperature on the rate of respiration in germinating seeds. Use the apparatus in figure 2.1 in your plan. So if you're going to ask use this apparatus, please look at figure 2.1 and then talk about this apparatus. Now, what are we going to do? At least three different temperatures below 70. So you said 35, 40, 45, 50, 55. I say use five temperatures. Use a water bath to maintain the temperature. Then give time for the apparatus to come to the temperature before taking the measurements. Then adjust the drop of colored liquid to beginning of scale before recording, which is like you say, reset the apparatus back to 0. At each temperature, you record the distance moved on the scale with time, then the same mass of seeds, then repeat and calculate the mean and calculate the rate for each temperature. And then you give me some conclusion. Increasing temperature increases the rate of respiration, or you can say increases till a certain temperature and then starts to decrease. Because you see, any graph, when you have temperature on the x-axis, the graph for temperature, if it's an enzyme control reaction, is going to be like this. So if you took temperatures which were less than the optimum, like you took four temperatures or five temperatures which were less than optimum, and say the optimum was 40. So temperature in degrees Celsius and temperature, you took only 20, 25, 30, 35, and 40. So you're going to get a linear graph. But if you took temperatures like 40, 45, 50, 55, then of course you will get points on the axis would be here, 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 or here. So you have to see is what temperatures did you take. So the conclusion usually is that if you took temperatures below the optimum, then of course you would see an increase in the rate. But if you took temperatures above the optimum, some below the optimum and some above the optimum, then you would get sort of a different readings. So how would you write this? It's very important that you write it very correctly. So five temperatures, I've stated the temperatures, but you could have just said, okay, take five temperatures. But I always feel stating the temperatures makes it sound better. We in fact give you another mark for that in your A-level exams. So how would you maintain the temperature? Use a water bath. The ones which I've underlined, you can write nearly in every planning question. Allow time for apparatus to reach a specific temperature. This is called allow equilibration time. You can also write this in nearly every planning. Set the color liquid to 0, or you can say reset the apparatus or starting from 0. Then at each temperature record distance moved by the colored liquid, then same mass of seeds, then repeat and calculate mean. You can write this nearly in every planning question. Repeat and calculate mean. Do not write average. It's repeat and calculate mean. Then calculate the rate for each temperature. This is important that you talk of the rate. And then conclusion, as temperature increases, rate increases until a certain temperature and then starts to decrease. Will it depend upon what temperatures you took? So this would be your 6 marks question, which is a long question. And some of you really find it very difficult. But I guess if you do about 5 papers, you then know what you've got to write. Question number 3. Figure 3.1 shows the whole of one seeded fruit of a dandelion flower. A single dandelion flower head can produce up to 200 of these one seeded fruits. The mass of a single fruit is 0.0005 gram. In the space below, make a large drawing of the whole fruit as it appears in figure 3.1. Now the diagram should proportionately be right. The seed and the stalk was longer than the seed. So you shouldn't have made it, you should proportionally make it correct. So whole fruit is drawn sharp pencil, clear and continuous lines, no shading. So you got 1 mark for that. Then 80 millimeter minimum length. So it has to be a big diagram. 80 millimeter is just for the examiners to mark it. If it was a very tiny drawing, if it was only a very short drawing, something like this, then of course this is very small, it's not 80 millimeter. So 80 millimeter length of the stalk and the seed. Then proportion stalk should have been drawn with a double line. This was a double line. The stalk should have been drawn with a double line to the top and longer than the seed. So this is the seed. So the stalk should have been longer than this. Detailed seed delimited and tapered and hooks drawn with double line. So these hooks should have been drawn with a double line and hooks only on the top half of the seed. So this is how this was going to be marked for 4 marks. Then it says the lines C and D indicate the total length of the seed and stalk. Draw a straight line on figure 3.1 to join C and D and measure the length of the line and record it. So where was C and where was D? Let's look at the diagram. So this was C. So from here to here, this is C here, and this is D here. So you have to measure this whole thing from here to here. And this was about this is about 36 millimeter. And then it says calculate the actual length of the stalk and record it to the nearest whole number. Some of you don't read that either. So what was the magnification? The magnification was 3 times. So this was very easy, 36 divided by 3, and the actual thing was 12 millimeters. So 36 divided by 3 is 12, 12 threes are 36 if you know your tables. So the actual length was, and it was the nearest whole number. Even if you had some other figure, you would have to give the nearest whole number. And the last part of the question, identify 2 features of this fruit that show it is adapted for dispersal by wind. Explain your answers. They're very easy. It's a low mass. It's very small, easily carried or picked up by the wind. And hairs increase the surface area, or you can say act like a parachute and helps it to glide. So low mass, small, easily carried by wind. Hairs increase the surface area, helps it to glide. That completes this paper, and thank you very much, and thank you for watching, and thank you for subscribing, and all the very best.